the AFO cafes. Once again this year, we have been working on a lineup of speakers, so I hope that you enjoy this talk. For those of you who don't know us, we are a professional ornithological association that focuses on studying and conserving birds and the natural habitats. And one of our strongest focuses is on Latin America. And we take pride in being a bridge between professional and avocational ornithologists. So be it if you're a birder or a bird lover, or if you come from the private sector, or do some other job that entails working with birds, you are welcome to join AFO. As I mentioned, we have a strong focus on Latin America through our outreach and scientific meetings, especially joint meetings, and we'll talk about that a little bit later, and also our grants programs. Given that we focus on science, I think it's important that you know that we have our own peer-reviewed journal, the Journal of Field Ornithology. You don't have to be a member. We are a membership association, but you don't have to be a member to publish in the journal. Our journal is open access. It is relatively cheap publishing it, and we are really glad to be able to offer either full or partial waivers, publication waivers under some circumstances, as well as offer a completely free editorial language assistance program for people who may have issues with language and need their some assistance with that. So if you have a research that needs publishing, make sure to check out the Journal of Field Ornithology. At AFO, we also have quite an extensive grants and awards program, and that is its primary end is to study and conserve birds and natural habitats. And we have 3 grants programs. One of them is for North America-based researchers. The other one is for Latin American-based researchers, and then we have our biggest grant, which focuses on or aims to actually provide funds to study little-known species in the tropics. So again, if you are a researcher, make sure to check out our grants programs. One of the things we also do is organize different events, such as these AFO cafes, which we do in order to be able to have a place for the ornithological community to, get together and interact, which is always a good thing, as well as organize science conferences, normally either annual events on our own or joint meetings such as the ornithological conferences of the Americas, which we hold every 4 years or so in a Latin American country together with ornithological NGOs from Latin America. As I mentioned before, we are a membership association, and we provide our members with different benefits, and some of them are access to our grants and awards programs, discounts on the events registrations, access to our Zeiss VIP program, which gives you a substantial discount on up to 3 items on the Zeiss Optics store, and free social media promotion, and the ability to join any committee that you want on AFO and have a chance to chip in and participate with us. I have a reminder here to let you know that the applications for the SCATCH research grants are open. These grants are aimed at Latin American researchers and also aim to promote question-driven research. So if you need some funding for your research, make sure to check out the link that you can see there that you can find later on the YouTube's description, and see if you can apply for it. The deadline is June 15th, so right on time before fieldwork season for Latin Americans. We also want to invite you to our coming conference in Ogden, Utah. We are very well underway the organization of this conference. Some important dates that you might want to have into account is that the abstract and travel awards deadline is April 7. We actually extended that date. We will be announcing workshops next week, and registration is already open with early bird prices going until May 16th. So if you want to come to the conference, make sure to buy your registration before May 16th to get the early bird price. We are also really proud to present our 3 plenary speakers. Gail Patricelli will be talking about robots, telemetry, and the sex lives of wild birds using technology to study courtship and conservation. We will also have Kini Roesler from Argentina, who will be sharing from the Hooded Grebe Project to the Patagonia Program, scaling up conservation efforts through flagship species, as well as Mark Hoiberg, who is one of our co-EICs at the JFO at our journal. And he will be giving a talk on learning from the birds across 6 continents, species recognition in brood parasitic cuckoos, cowbirds, and their hosts. We are really looking forward to this conference. Ogden is beautiful. This will be a solo conference, and we will have plenty of field trip options, and the landscape around is just beautiful. So we hope that you can come and join us. Finally, if you want to keep up to date with our activities, make sure to follow us either on Instagram or Blue Sky as at Field Ornith, or in Facebook as the Association of Field Ornithologists. And if you want to learn more about our association, check out our website, afonet.org. AFO Cafés are sponsored by AVNet Research Supplies, which is the business venture of AFO. AVNet provides high quality research gear and ethically and handmade mist nets. So whatever you need for your research, be it blood sampling or tissue sampling stuff, be it gear, field guides, mist nets, or whatever, make sure to also check out avnet.com. I forgot to add the, the link, sorry, but it's avnet.com. Finally, our next AFO Cafe is still to be defined, but if you enjoy this cafe, please consider becoming an AFO member. You would be supporting these and actually everything that we do. And if you want to nominate yourself or someone else to speak at a future cafe, please send me an email at agustinatorreta@afonet.org. I will leave you the email on the description as well. All right. I will pass on the mic to Juan Manuel, and he will be presenting our speaker for today, Alex Young. I will make just a very small clarification, and I communicated this through socials, but just so you know, Alex will be giving his talk in English, and the slides will be in Spanish for more accessibility for the Latin Americans joining today. So without further ado, Juanma.
Thank you, Agus. Thank you, Agus. Well, we're greeted to present today's speaker, Dr. Alex Chan, who happily and almost immediately accepted to participate. Alex's research focuses on the impact of rapid environmental change on birds and how different movement strategies allow or constrain their ability to cope with change. Since his PhD at Florida's University on neotropical tyrannid migration, Alex has migrated over many other taxa, collaborating with colleagues in Argentina and Colombia, He has published more than 60 academic papers, presented numerous conferences, and also participated on the Neotropical Outreach Committee of the AFO. He has recently started collaborating with local partners in Argentina to study how nomadic South American flamingos move through the year and how they're being impacted by climate and habitat change. I personally know him from his extensive research and also extensive fieldwork on the scissor-tailed flycatchers from El Destino Reserve. His commitment and his endurance have been an inspiration for me and for everyone who had shared fieldwork with him. And so, Alex, now it's up to you.
Thank you, Juanma. That's a great introduction. I really miss my Argentinian friends when I'm not down there. And it's great to see a lot of you here today attending. I'm going to try to share my screen here. Let's see, I'm going to replace. Okay, and see if this works. Okay, does that look good? Okay. Well, yeah, it's really exciting for me to talk to you all today about this project on these incredible birds. I only started studying flamingos a few years ago when I was at the Smithsonian Migratory Bird Center, which had funding to cover the start of this research. And really, this research isn't possible without the strong collaborations that I've developed with researchers from across the hemisphere, Argentina, the United States, Chile, different places where these flamingos live. And yeah, basically this type of work, this large-scale project is impossible to do without very strong a network of collaborators. So thank you to all these people on the screen and even others that we're going to hope to start collaborating with soon. And just a quick background about flamingos. There are 6 species of flamingos on the planet. Four of these species live in South America. So the 3 at the top of the screen, the Chilean, the Puna, and the Andean flamingo, they are all spending the summers in the Andes, as well as in, some of these have populations in the lowlands as well, but they're really well known to go uphill into the Andes during the summer, this brief summer period of hyper productivity in these lakes. And then the American flamingo there at the bottom of the screen occurs in the Caribbean, throughout the Caribbean, and then in northern South America. But I'm going to just be talking about the 3 species of flamingos there at the top of the screen today. And these lakes, which some of them are located above 14,000 feet in the Andes, are very cold and dry for most of the year. They become salt flats. And once these rains start falling in the summertime, beginning around November, December, they just become these hyper productive lakes that are kind of like the Arctic in the sense that it's a very short summer, but it's very, very high productivity of zooplankton, phytoplankton, all these microorganisms that these birds are looking for. And these lakes are located mostly in central South America, where these birds spend the winter. On the left side of your screen, you see the James flamingo. Then in the center, the Andean flamingo. And there on the right, the Chilean flamingo counts. So these are data from the High Andean Conservation Group that's been collecting this data for over 20 years. This is the latest data that's set from 2022. And the larger circles basically show where most of these birds are hanging out during the counts, which are done in February, late summer. And as you can tell from the numbers, the Chilean flamingo is the most abundant species, followed by the James flamingos, and then the Andean flamingo there in the middle is the most kind of rare, the most endangered of these birds in terms of numbers. There's an estimated global population of about 80,000 Andean flamingos right now. And the threats that these birds are facing in the 21st century are a multitude of threats, but what has been identified so far is that climate change is happening very rapidly at these high altitudes in the Andes. So that's a main factor. And the second is the rapid change in the habitats, a large-scale habitat change at these remote sites where a lot of mining is happening. And the most recent type of mining is lithium mining, of course, but a lot of other different types of mining have been happening for decades, even centuries in these parts of the Andes. It's just getting larger scale now. In terms of lithium, over half of the world's supply of lithium is found in the central Andes, primarily Chile, Bolivia, and Argentina. And as you know, with this push to develop electric vehicle technology, these rechargeable batteries are needing lithium. They're also in our phones, and any battery that needs to be recharged needs lithium. So there's a very large market demand for this element. And the so-called lithium triangle just happens to be located right in the middle of the central Andes, in the middle of the distribution of a lot of these these three flamingos. As you can see on the right-hand side of the screen, the distribution of the Andean flamingo almost perfectly overlaps that of the lithium triangle, where again, most of this lithium is found in these salt flats, shown in that kind of tan color there. So that's a major concern. The problem is nobody really knows how mining or climate change or just the rapid transformation of the Andes, we don't know exactly how that's impacting flamingos or biodiversity in general at this part of the world. So it's a major gap in knowledge. As you'll see in this talk I'm giving today, we're just trying to get a foothold on the basic uh, patterns that are happening and the baseline of how these impacts might be translating to impacts on the flamingos. So we're tackling three main questions. The three big questions that we have in this project is which wetlands do these birds use at different times of year? Second, how do they use each wetland? Does their diet vary, depending, is there different food availability and types of phytoplankton, zooplankton in different types of wetlands, depending, for example, on salinity? So we just have to get a basic understanding of the ecology of these birds in different wetlands in different seasons. And finally, what are the risks that these birds are facing in different regions of their range and different wetlands? So to tackle the first question, we basically have to develop a connectivity map. So how do these birds move between wetlands? Are they, using specific wetlands at one time of year, and then how do they move to these other wetlands? That we know that they move a lot. For years now, people have known that these are altitudinal migrants. They primarily spend the summer at high altitudes and the winter at low altitudes. It varies a little. Andean flamingo, I'll show you some of our preliminary data, but Andean flamingo moves in different ways than Chilean flamingo, for example. So there's a lot of variation, and it's more of a nomadism than a migration because their movement patterns between different wetlands is not very regular. In migration, we think of back and forth on a regular cycle, whereas in nomadism, it's hard to predict where they're going to go next. And that's more of what we're seeing. But to answer this question, we basically have to go out there to this area, this large scale. It's a large scale problem because we're talking about an area of hundreds of kilometers large. So for example, in this Andean flamingo range, how do they move across this large range? And within this lithium triangle, it's a problem of several hundred kilometers in the sense that, they are, let's see if I have I think my screen froze for a second. Let me see if I can get my cursor to move. Okay. So I've overlaid the state of Oregon's outline on top of the lithium triangle area here to give you an idea of the sense of the problem. At least for people in North America, you can understand. Oregon is a very large state. It's about 500 kilometers north to south. So these birds are moving across a very large area. But it's a very beautiful, spectacular area, as many of you know who have visited, full of volcanoes. These are vicuña standing in front of a volcano near to one of our study sites. There's a lot of wildlife, a lot of wild places. And that's, these are places that haven't seen any human development in any time in their past. So all this change is happening very quickly in very pristine ecosystems that have never experienced this kind of change. And this is a landscape also that's been inhabited by Native Americans for hundreds of years. As you can see this archaeological site here next to Lake Vilama in Jujuy province of Argentina. It's a protected area, but it's apparent from archaeology that people have been going to Lake Vilama for at least 500 years to harvest flamingos every summer. So flamingos, people, and the ecosystem have been interacting in this very remote regions for a long time. And now we're interacting too in terms of research. Fortunately, we don't have to live under very primitive conditions there at Lake Vilama. Working with Enrique Derlindati at the University of Salta there, we have access to these biological station that's basically shipping containers that have been placed there through collaboration with the university and the province. So that helps a lot. It gets really, really cold at night, as you can imagine. That's at 14,000 feet elevation, about 3,500 meters above sea level. So it's really extreme conditions, even in the summertime. And how do we catch these flamingos? Because to understand this connectivity, we want to put satellite transmitters. We're using satellite transmitters to track their movements. Well, these are basically leg hold traps that are under the water. And every 5 or so meters, we have a slip knot that when the bird steps into it, it closes on its leg. And then we quickly extract the birds. We're standing there next to the shore watching these traps constantly. And once we have the flamingo, then we just take it to the shoreline. And just like banding a songbird, we put a band on its leg and then take a series of measurements, standard banding measurements on aging, sexing, taking morphological measurements, weighing flamingos. Of course, you have to use a pistola with a bag, and then we take a series of pictures. Nobody even knows the molt schedule of these birds, so we're trying to understand what the molt schedule is like, understand what full adult plumage looks like, what differences are there between the sexes and even between populations. And then right before we release the bird, we take some physiological measurements. We collect blood, for example, for glucose, ketones, triglycerides, hemoglobin. We can measure all these now in the field with those handheld meters you can see on the screen. So it's really nice to have this technology that's readily available. And so we take this, and for genetic testing as well. And then right before we release it with a satellite transmitter, we are outfitting these. These are GPS Argos tags that allow very precise tracking of the birds through GPS technology. So we can get with a resolution within just a few tens of meters. On the right there, you can see a map of several of the flamingos that we're tracking, just to give you an idea of what the data looks like. But we're just starting now to work with colleagues on the processing of this data. Jorgelina Asmus is going to do her postdoc in Argentina using this data for her postdoc. So we're excited to start understanding basically what these data represent. Just really briefly, some preliminary data we have from Andean flamingos shows that there's a lot of variation between seasons. So you can see there on the left-hand side of the screen, the different seasons of the year. So from October to November, we're seeing the highest movements. So that's spring migration, basically, or spring movements uphill from the lowlands into the Andes. That movement persistence is just a value that gives you an idea of how much that bird wants to move day to day. So not surprisingly, in the spring, they're moving more than in any other time of year. The altitude that they reach is almost 4,000 meters above sea level in the summertime. Again, that's December to February. And then They go, most of these birds, not all of them, but most of the Andean flamingos we've tracked, and only about 6 or 7 so far, they've gone down into the lowlands. Some go to Bolivia, as you'll see in a little bit, to the Altiplano of Bolivia, which is lower elevation at 12,000 feet for the winter. But the highest elevation there reaches in summer. And then there in red, the rate of movement, kilometers per hour, it's highest in right there in the fall. So what's probably happening March to June is they're starting to search for a place to spend the winter. They're going downhill into the lowlands and they start foraging, recovering maybe from breeding all the calories they've spent during the breeding season. They have to recover those. So their movement goes up and then it goes way back down again during the middle of winter. And when they're in the lowlands in July, September, it's the lowest there. And yeah, we have, I think that's all the data for that. And then this is just to give you an idea of the tracks that we're getting for some of these birds. So these sites in the Andes, SL, LVP, LP, those are just abbreviations for these lakes. LVP is Laguna Vilama, the lake I showed you pictures of a little earlier. That's the main site we've been banding at. And as you can see, this one, there's 2 flamingos here, Andean flamingos on this map. On this slide, the map on the left shows an Andean flamingo that it kind of moves between Laguna Vilama and then LMC there on the lower left is Laguna Mar Chiquita. Mar Chiquita is a very large lake in the lowlands of central Argentina at sea level almost. And a lot of the Andean flamingos and James flamingos that we're tracking, they go there to spend the winter. And so this particular individual in green on the left side of the of this screen is moving between Vilama, Mar Chiquita, and some places also within Salta province. And the bird on the right seems to be moving exclusively between Laguna Vilama, Mar Chiquita, and Laguna Popo in Bolivia. That's LP. So I believe in one year it went to Bolivia and in another year it went to Vilama. And to give you an idea of the resolution within each lake, we can also understand kind of how are they using each lake. So here are 2 flamingos in Laguna Popo, Bolivia. One is using the northern part of the lake, kind of it's hanging out in the middle of the lake, and the other one is in the southern part of the lake, but it's even going into streams, and there's a small stream that empties into the this southern part of the lake, and this bird is actually using parts of the stream as well as the lake. So it's moving between the shoreline and the stream. So we can model habitat use with this technology as well within lakes. And this is just a GIF to show you the movements of several of these birds that you can see different. Each color is each point that moves that's a different color is an individual bird. This is a combination of the 2 species, James flamingo and Andean flamingo, but they move in very similar ways. The take-home message here is that these birds are moving very rapidly. It only takes them a couple days to move 1,000 kilometers. They don't stop. There's nowhere to stop between lakes. It's just desert out there. So when they move, it's very quickly. There's hardly any stopping over between sites. They move to a new site and then they stay there for a long time. It might take them 2 or 3 days to move 1,000 kilometers, and then they'll typically stop for a month or 2 at that one site. Once in a while, you'll see them stop for 2 or 3 days and then keep moving, but it's not as common. And as you can tell, I mean, there's no pattern to these movements. They're just moving. You can see the date changing at the top of the screen. But and here it's entering December or January. They're up in the highlands mostly. But some are staying in the lowlands. There's a lot of variation between years. There's no distinct pattern to movement. So that's why it's mostly nomadic. That's the best way to characterize their movements at this point. But let's see if I can get this to show. There's map layers that we can use to overlay their movements. So this is a temperature graph, for example. So one of the main challenges next, now that we have raw data coming in from the satellite tags, is to overlay layers such as precipitation, temperature, maybe even habitat change through the mining consortiums, where are they located, to see how are the movements of the birds, how are those movements a response to these different environmental parameters. So that's the first part, is basically this tackling how they move between sites and how are they using different sites at different seasons. And then that gives us the ability now to start understanding how do they use each site. So when I say how do they use each different site, we basically want to begin by understanding their diet. There's some basic descriptions of flamingo diet already, but it's a very baseline description, in the sense that we don't understand how each of the three different species, Chilean, James, and Andean flamingos, how do they forage at different times of year? Do they compete for food? We're not really sure. What is known is that these are a picture there, the image there on the left-hand side of the screen shows just how many microorganisms. I just took that with this picture with my phone. It's the water just teeming with brine shrimp, and then the water turns pink basically from the flamingos pooping into the water after they've eaten. It's basically an ecosystem that's dominated by these birds in terms of their almost potentially ecosystem engineering that they're doing. And this is a picture again of just how a scoopful of water shows you how much the food there is in this water during the summertime. And those lamellae there on the right-hand side of the screen, that's a close-up picture of the bill of the flamingo. Those lamellae allow it to filter these plankton out of the water. So to understand how much food is available, first of all, here's a picture of Nico and Pamela sampling the water, and we're filtering using plankton nets here to take the samples back to the lab and then characterize using a variety of techniques. We're going to try to use metabarcoding, for example, to understand how these birds are in their diet. How are they competing potentially for food? Do the three different species forage on the similar prey items? We're not sure. That's one of the main questions. And we also take pictures of the bill, standardized pictures with a ruler to use on-screen, image processing to understand how does their morphology match their diet. So the third question about how the risks to these birds change throughout the year and between sites is basically requiring environmental sampling. So here Nico is taking a swab of feces that he found on the ground. We sample the water, we sample feces that we collect from the birds as we're banding them, and at these foraging sites, you can see the flamingos there in the background. So just basically collecting environmental DNA. And Nico is also doing a project on microplastics. So within the water samples that are collected, searching for types and amounts of microplastics in the water. Believe it or not, these sites are still very remote, but there's evidence of microplastic contamination within some of these places. So that's one of the main challenges, as well as understanding their exposure to heavy metal amounts in these different lakes, either naturally occurring heavy metals, or naturally occurring, or also as a result of anthropogenic change. Let's see, I think my screen froze again. Okay, and yeah, this is just a picture of the types of samples that Nico collects, and then we're taking them back to the lab right after the field sampling to spin sometimes using a centrifuge, and in this case using a vacuum technique to separate the water from the sample and to purify it as much as possible for mass spectrometry. And one of the main challenges also is understanding how does a flamingo basically tell you the condition it's in. So is there a way we're trying to develop a quick and dirty way to sample flamingo health without having to collect genetic, heavy metal, all this complex data sets on each flamingo, which it's going to become very expensive. So one of the questions we're asking is there a way to understand how these flamingos communicate to understand their health, their exposure, for example, to heavy metals? So flamingos are famous for doing these dances where they are probably showing each other, their abilities, either their different morphological variables that they're showing off as a signal. And one of those obvious ones, given their coloration, is the plumage color. So we are using a spectrophotometer to collect color data for each flamingo that we capture. There on the left, we are collecting data on the color of the bill. On the right is collecting plumage color data. It's a very simple technique in the sense that it doesn't collect data in the UV spectrum, ultraviolet. It's just a visible color because flamingos, from at least in the literature, what's known about them is that they are principally, unlike songbirds, they're principally perceiving light in the visible spectrum, just like we do. And the really interesting thing that's come out from recent research in Europe on greater flamingo is that they're apparently capable of using cosmetic coloration or makeup using the carotenoids in the uropygial gland oil. So the carotenoids that are found in this zooplankton, phytoplankton that they consume, making their feathers pink when they molt them, also makes their uropygial gland oil pink to a certain extent, not as pink as the feathers necessarily, but what they found with the greater flamingo in Europe is that the preening oil, this oil that they use to waterproof their feathers, actually turns pink, and as they apply it, it makes the feathers even pinker. So they can become pinker overnight, practically as they preen. And this is basically what we're attempting to document in Andean flamingos to see if it applies, because if it does, then it's the coloration that we're getting here in the plumage is actually a direct measure of their current ability to consume phytoplankton and the carotenoids that come with it. So if we can measure this color in real time, we're getting basically an idea of how good their diet is, in terms of how good in quotation marks, because there's a lot of ways of measuring quality of diet, but the general understanding is that the more carotenoids that these birds have in their diet, the better for them. Carotenoids are an antioxidant, for example. And so there's a lot of different ways of measuring color. This is the color output values that we get from that spectrophotometer. So this is a color curve from one flamingo that we get. So we get a lot of indices down there at the bottom, for example, like RGB. But this color spectrum there at the top of the screen is actually the most valuable because it gives us values across the visible spectrum from 400 to 700 nanometers. There's three main measures that scientists use to measure color, and that's tone, value, and chroma. So if you're familiar with this color system, it's called a color wheel. Tone is the value of the color. It's just the colors that are located around the wheel. So red, yellow, green, blue, pretty basic. And as you go up and down that wheel, you can see there's a cone there in the middle from 0 to 10. That's just the lightness or how light or dark that given color is. So you could have dark red or light red, for example. Light red being at the top and dark red at the bottom. Chroma, there is more of the horizontal value from 0 to 12. So the chroma is the value, how rich or intense is that color. So value, hue, and chroma together, if we can measure them using this spectrophotometer and we take them together, then we basically get an idea of the the quality of the color that each bird, each flamingo that we capture is producing. And once you can measure these three values on this color wheel, you essentially get an idea of just how how much quality diet this bird is consuming. At least that's what they found with greater flamingos, for example. They found that flamingos with more higher fat deposition were a deeper red color. And yeah, we're getting some preliminary data from the carotenoid chroma. Lin Wang is helping us finish up this part of the research for now, but we have this preliminary data from a student I worked with while I was at Indiana University. So the axillary feathers that are under the wing are the richest red, is what we've found so far. And then the color on the neck and the head is the lowest, has the lowest values. And this could change. The more data we get, the more we'll understand how it varies amongst ages and sexes and maybe even populations. And finally, we shouldn't forget that this region, that the Central Andes, where we're studying flamingos, it has been and it continues to be inhabited by people who share the ecosystem with these flamingos. They have many of the same needs. They need clean water, for example. They understand, they kind of grow up with these flamingos practically in their backyard. We were banding flamingos one day near a community, and there was a grade school right there next to the lake. So the kids came out and watched us band the flamingos. We're giving talks at their schools to, they ask us, what are you doing here? They're really curious, they're interested. And so we tell them what we're doing, why it's important to conserve flamingos, and we're learning from them as well, and they give us their artwork, how they interpret the flamingos, and so it's great working in these communities. We're developing a lesson plan where we take this foldable microscope made out of cardboard that can be attached to an iPhone, Samsung, it doesn't matter which type of phone, but it basically attaches to the camera on the phone, and then it becomes a microscope so that the water that you put under that microscope can be used right there in the school. Believe it or not, even out there, way out in hundreds of miles from the nearest city, people have smartphones. There's technology everywhere these days. So that's one of the lesson plans we're developing there with the school teachers in Argentina. Finally, just a general message we're sending to researchers, community members, stakeholders, is that both humans and flamingos need the same things to be healthy, and one of those is clean water. So a healthy ecosystem is good for everyone, is our main message. I'd like to thank our funding for this project to date. And as we collect more of this data, we hope to really turn these flamingos into sentinels, true sentinels of the health of these amazing ecosystems. And if you want to follow us as we collect more data, we have an Instagram group called Proyecto Flamencos Sudamericanos, with an Instagram page. So that's the link. And fortunately, the students there at the University of Buenos Aires are managing that, keeping everyone up to date through that. That's the best way to keep up with us. And of course, if you have questions, happy to answer them through email as well. Happy to answer any questions now. Si tienen preguntas, también puedo responder en español y traducir. Thank you very much. Okay.
That was so good, Alex. Thank you so much. That was so interesting. I see everyone is reacting with claps. Well, Juanma has raised his hand, but I do have a question for you. And I apologize if it sounds kind of silly because I'm not an academician, just a very avid birder. But I wanted to know what kind of information do you get from the blood samples that you take? You mentioned that you measured like the triglycerides and other components. What kind of input do you get from that? Like, is it overall help?
Yeah, so for example, with triglycerides and glucose, it'll give you an idea of the calorie intake of that bird and at that moment. Well, triglycerides more the fat, the kind of lipid, that lipid uptake that they're getting. But you have to combine that with other measures like classic body mass, you know, understanding how much fat deposition these birds have. Um, we also collect the ketones values. So typically ketones are a measure of how much of the bird's own reserves it's burning, right? Is it burning fat or is it getting most of its calories from the current diet? Um, but the other measures of the from the blood we're getting are understanding their immune function through the blood slides. So we'll take a blood slide to get counts of white blood cells, red blood cells, and that'll give us basically you know, you can even measure some diseases through the blood slides, but it's mostly an idea of red blood count, like hematocrit and immune function. And we do spin the blood when we're near a centrifuge. We try to collect. We try to spin the blood for hormones. So stress hormones, corticosterone, is a big one to understand how stressful is different are the different times a year.
Right.
Yeah, and then we haven't done any genetics yet, but we've been discussing some more molecular research with Kevin McCracken's lab at the University of Miami, where he has a lot of experience with molecular techniques and diving ducks in the Andes, for example, and applying some of those techniques to flamingos. He wants to study, for example, mitochondrial respiration. So how is mitochondrial respiration at sea level versus at high altitudes at different times of year?
So interesting. Thank you.
Thank you.
Does anyone else have any questions? Feel free to turn your cameras on if you want for the discussion. Juanma, you want to say something?
I was actually clapping, but I was wondering if you expect to find some kind of genetic structuring between the salt lakes you were sampling, or if you expect that the movements are kind of temporary, or you might expect some kind of genetic structuring, flamingos that happen to be sampled in some place remain there.
Yeah, that's a good question. I think it's going to depend on the species, because, for example, Chilean flamingo is widespread from Patagonia.
Mhm.
You know, they have breeding sites in southern Patagonia and Santa Cruz province, all the way up into Peru. So I wouldn't be surprised if there's some major genetic structuring across that range, that large range where Chilean flamingos, you know, we don't even know if Chilean flamingos do they leave Patagonia at any time of year? You know, what kind of gene migration is happening, like gene flow between populations of Chilean flamingo. We've talked about you know, potentially putting satellite transmitters in different populations, starting with Chilean flamingo because they're so common and abundant. That would be probably the best species to start with if we want to understand just how much gene flow occurs in a flamingo population. But you know, as I said earlier, Andean flamingo is the most probably threatened of these three species because there's only 80,000 Andean flamingos. So I think understanding inbreeding, for example, in that species is going to be very important. Um, you know, how much does inbreeding impact these birds? So I wouldn't be surprised if we find very low genetic structuring between Andean flamingo populations, just because they move so much. They move a lot, and their range is much smaller than Chilean flamingo. So, but I'm just guessing here. I'm not sure. We're working with Juan Carlos's Reboreda's lab, Nico Lois, Kevin McCracken. We've been having discussions about collecting more blood across different populations to start a more broad scale population genetic study.
Alex, we have Dale raising up his hand, but Dale, if you don't mind, I'm going to first read a question from Manuel, who left a question in the chat that says, "Hi Alex, I love the talk. I am very interested to know if you have any information whether you have significant differences between the three species of flamingo." That's Manuel Gabo.
Okay, yeah, in the movement, we do. In the movement patterns, we're seeing Chilean flamingos are moving much less than Andean flamingos. Andean flamingos seem to move the most. And we're not sure exactly why. It might be because they have a more specialized diet. That would be a hypothesis. So Andean flamingos might need to track specific food resources more throughout the year between lakes as they become available. Whereas, if Chilean flamingo is a generalist, if they're more of a dietary generalist than Andean flamingo, you might not need to move as much. But that's just, like I said, it's a hypothesis. I suspect that differences between movement patterns between species as well as between intraspecific populations will probably, it'll be diet driven by how specialized that diet is.
Great, thanks, Alex. Go ahead, Dale. Sorry.
No, that's all right. Alex, great talk. Really neat system. I love the system. You mentioned you were kind of interested in maybe competition between the two flamingo species. Are there any other water birds that use those systems, maybe on migration or other times? What are the other water birds out there, I guess,
Yeah,
is the
that's a
question.
really good question. And it's just amazing, but there's only one other bird that we've seen doing any sort of feeding in the water for, plankton. Well, to begin with, these are hypersaline lakes. I can't emphasize hyper more than anymore. It's off the charts. We put a probe in to measure salinity and it's off, every time it can't measure it. So we have to dilute the water. It has to be diluted several times, like several orders of magnitude just to measure it. So there's basically nothing else in the water except plankton. There's no fish, there's no amphibians. And the only other bird species we see foraging alongside the flamingos are Wilson's phalarope. So they arrive, about October. And it's really cool because they many times are hanging out right at the feet of the flamingos, almost taking advantage of the flamingos foraging to get their food. Where it'd be really cool if there was some sort of a symbiosis there. I'm not sure, but yeah, as you can imagine, these phalaropes just arrive by the thousands. And as far as we can tell, it's the only other bird in the water with them. I don't think anybody else can take that level of salinity. There's Andean ducks nearby, but they're not in the lakes. They're all the other waterfowl, like the ducks, the Andean goose, they're hanging out in the streams and the rivers that go to the lakes. And these are endoeric basins in the sense that there's no there's no water flow through these lakes. It's just input from snowmelt and rain, and then it's just evapotranspiration. So any fresh water, all the fresh water is outside the lake in the streams and springs.
Thanks.
Much more saltier than seawater.
What about the avocets? Do you see avocets feeding next to the flamingos?
Yeah, we have seen avocets. I think in Mar Chiquita we've seen more avocets than up in the highlands. Of course, in Mar Chiquita there's a lot of Wilson's phalarope as well, but there are avocets in Mar Chiquita. I think there's just more diversity in general in Mar
Yeah.
Chiquita. We have been sampling the water in Mar Chiquita, and it's such a large water body that
Yeah, it is probably more stable, right?
yeah, it's probably more stable than these lakes in the Andes. Vilama is about 16 kilometers long. It's fairly large, but not near as big as Mar Chiquita. You can't see the other side of Mar Chiquita. So there's probably a lot of variation within Mar Chiquita. There's probably like more salinity in some places than in others where maybe the avocets can go. But they're still, we're talking about a few hundred avocets, but thousands of flamingos. So
Yeah.
yeah, right now with all the rainfall in Argentina, as many of you probably know, we're going through a very strong El Nino right now in the southern hemisphere. There's flooding and disaster in many parts of South America. So a lot of these lakes are flooded right now, and we just saw one of the Andean flamingos just go to Vilama for just 3 or 4 days, and then it left. Vilama a couple of years ago was flooded, and the flamingos had to swim. So there was not enough, like there was no place to stand in the water. It just the shoreline went down so quickly that they have nowhere to stand in the water. They'd have to forage. They basically have to do this dipping that you see geese. They kind of dip into. They have to put their entire front of their body into the water, and so that's what the flamingos ended up doing as well. They have to almost swim. And so too much rain is probably stressful as well as too little rain when there's no water. So either one is probably stressful for them.
Very interesting. Do you have any more questions? Anyone else have any other questions for Alex? We had a comment from Dot who says, "Great talk, Alex. Love the smartphone microscope attachment and of course the flamingo drawings."
Thank you. I'll tell the kids to keep drawing.
Do you know if the local Andean communities have adopted flamingos as part of their mythology? They have?
I'm not sure about that. I've asked around a little, and beyond their dependence on flamingo eggs, especially, they would harvest the eggs
Yeah.
You can imagine it was a major protein source. These eggs are large. I haven't seen any evidence that they had, they didn't leave much evidence behind anyway. But as far as I know, we don't know of any kind of how they perceive the flamingos in terms of their worldview. But I wouldn't be
Right.
surprised maybe in Peru, maybe in other countries. I'd be interested if anybody knows about that, if there's evidence for how flamingos were perceived or by the Native Americans. I would appreciate information on that.
All right. Do you have any more questions, comments for Alex about the AFO Café? Are we ready to go home? All right. Matt, should we wrap it up?
I think Manuel is raising his hand.
Oh, okay. Manu, go ahead.
Hola Alex, ¿puedo preguntarte en castellano?
Sí, claro.
Ok. Viendo lo que estaban hablando antes sobre algunas diferencias entre las lagunas y antes Agus te preguntaba sobre la biodiversidad, pensando en las grandes concentraciones de aves migradoras que hay en Mar Chiquita, ¿no? En Córdoba. Me imagino que la biodiversidad o la disponibilidad de alimento por ahí para los flamencos también debe variar, ¿no? No sé si estuvieron o si conocen las la disponibilidad de alimentos, si varía entre las lagunas de altura, no sé, me imagino que va a haber diferentes especies de diatomeas, no tengo idea, ¿eh?
Sí.
Se me ocurre, por ahí, si conocen eso, me parece súper interesante saber si existen esas diferencias.
Yeah. So Manu is asking whether we have any evidence of how food availability differs between the high Andean lakes and the lowland lakes like Mar Chiquita, from those plankton samples we're collecting, for example. Manu, no tenemos evidencia todavía. Recién en el septiembre pasado durante la RAO.
If you want, you can
Okay.
answer me in English.
Okay, well, so we don't know yet. That's the short answer. We just started sampling the lowland lakes during September of last year, actually right during the RAO meeting in Cordoba. So we have just begun, but just by looking at the samples, that we've collected, it's very evident that there are going to be differences. It will probably, there probably won't be a very distinct difference between highland and lowland, but I think it'll probably depend on the year. For example, with this El Nino year right now, it's probably going to be a very extreme year. I would guess that there might be lower abundance with El Nino because of all the water. So it might be more diluted with more rainfall. Whereas maybe in a normal year where it's less rainfall, but not too dry either, there might be more abundance because there's just less waters. But I don't know. We're collecting pH, salinity, conductivity, turbidity to get an idea of how do all these parameters influence food abundance. But Nico in Buenos Aires, he's in charge of the water samples, and we're working right now on understanding, basically analyzing that to quantify those measures. So I'm sorry I don't have a more precise answer, but we're working on it.
Thank you, Alex. Well, we will wrap up this AFO Cafe with this question. Thank you all for coming. I hope you enjoyed it. Thank you, Alex, for saying yes, for accepting our invitation. It's been a pleasure. If any of you would like to nominate yourself or would like to nominate anyone else to do an AFO Cafe, we would love to hear your proposals. You can reach out through any social media outlet that we have. Just look us up as the Association of Field Ornithologists. And well, I hope that you have a good end to your week. And I hope to see you all soon. Thank you again for being here. It's been a pleasure.
Thank you, Alex.
Thank you.
It's great.
Bye-bye.
Thank you.
Thank you.
See you, everyone.
See you. Bye.